Hello friends, myself Professor Khemchand Surana. In previous sessions, we have started about the anti-amoebic agents. In that we have seen, in first session, we have seen about the protozoal infections, basics of amoebiasis and life cycle of Entamoeba histolytica. And into the second session, we have started with the classification of these anti-amoebic drugs, and we have seen that these three types of classification of amoebiasis. The first one is basic clinical classification, second one is based on tissue amoebicides and luminal amoebicides, and the last classification, which is what, chemical classification of anti-amoebic agents. Further, we have discussed about the drugs which were effective in both cases, which is the intestinal as well as hepatic amoebiasis. So we have started with the nitroimidazole and discussed the details of this metronidazole basically. So in this metronidazole, we have seen the basics of metronidazole, mode of action of this metronidazole, the chemical conversion of these metronidazole, which leads to the shows the anti-amoebic activity, and the metabolism of this metronidazole. Further, their side effect uses we have seen, and last one, the synthesis we have seen of this metronidazole. Further, we will continue with this class of nitroimidazole, which are what, the anti-amoebic drugs. So the first drug already we have discussed, which is metronidazole, and the second drug from this class is trinidazole. This trinidazole drug is chemically related to the metronidazole, so which is what structural similarity. So we can see the structure over here. 1,3 nitrogen is there, at second position CH3 is there, whatever at say first position CH2 S double bond O CH2 CH3, and at fifth position there is nitro group. So the IUPAC name of this trinidazole is 1,2-ethyl sulfonyl ethyl 2-methyl 5-nitroimidazole. So this is what structurally similar drugs from the same class. So it is prefer over metronidazole as it is more potent and longer acting and having less side effects. As already we have discussed, metronidazole, so this trinidazole is what having more advantages. So which is, this trinidazole is what, more potent than metronidazole, longer acting and less side effects. So also the metronidazole having major kind of side effects, so this trinidazole having a less side effects. So again the structure given over here, you can see an IUPAC name is also given. So the basic mode of action of this trinidazole is, trinidazole mechanism of action is similar to that of metronidazole. Whatever the mode of action of these metronidazole we have seen, that is what same mechanism of action of this trinidazole is there. So what is that basic mechanism of action we have seen? So whatever these pyruvate ferredoxine oxidoreductase system, which will reduces the metronidazole into the metabolite, and these metabolite will penetrate into the cell, and these whatever metabolite will causes the toxicity to the whatever amoebicidal cell or entamoeba histolytica. So this is what the basic mechanism of metronidazole and trinidazole. So it is used in the treatment of intestinal as well as hepatic amoebiasis with the potential greater efficacy than the metronidazole. So this drug is basically useful in intestinal as well as hepatic amoebiasis and having potential greater efficiency than the metronidazole, means more efficient than this metronidazole. So metabolism of this trinidazole is, which will metabolize by hydroxylation at 2-methyl group and catalyzed by the CYP3A4 to form inactive compound. So this is what the basic metabolism in metronidazole case. Also we have discussed the two basic pathway. First one is hydroxylation and second one is oxidation. Hydroxylation occur at 2-methyl position and gives hydroxymethyl metronidazole, and at oxidation, so metronidazole acetic acidyl metabolite will get. So here is again the hydroxylation is there at 2 position and again catalyzed by this CYP3A4 to form inactive compound. So this is what about tinidazole. The next drug is ornidazole from same category. Structure is again given over here. Imidazole basic structure. At first position CH2 CH2OH CH2Cl. Third position nitrogen. Fourth position is free, and at fifth position there is NO2. So the IUPAC name of this ornidazole is 1-chloro 3-2-methyl 5-nitro 1H imidazole 1-vial propane 2-all. So this is what the basic structure and IUPAC name of ornidazole. So mode of action of this drug is after passive absorption into bacterium cell. So initially what this ornidazole will passively absorb into the bacterium cell. The nitro group of this ornidazole is reduces to amine. So whatever this ornidazole having a nitro group at fifth position, which will reduces into the amine by this ferredoxin type redox system, and the formation of redox intermediate into the intracellular metabolite, which believes to be the key component of microorganism killing for ornidazole. So after this absorption, which will reduces this nitro group into amine, and this amine metabolite, which is what the key intermediate or we can call metabolite for killing of these Entamoeba histolytica or the bacterium species. So the drug is active against anaerobic bacteria basically. So this is what about ornidazole. This is the all category of this metronidazole. The first drug we have seen, that is what metronidazole, second one is tinidazole, and third one is ornidazole. So this is what the first category. Now second category from this chemical classification is dichloroacetamide derivatives. So in these dichloroacetamide derivatives, these are what the examples: dialoxanite furoate, etofamides, and tecloxane. So these are what the three examples of drug from these dichloroacetamide derivatives. So these drugs basically dichloroacetamides, which were effective in the intestinal or luminal or local amoebicides. So these drugs were effective in intestine also, luminal also, and local amoebiasis also. So the first drug from this category is dialoxanite, which is what directly amoebicidal agent, means directly amoebicidal agent. The mode of action of this dialoxanite, before the mode of action we will see the structure. The structure is given over here of dialoxanite. The furan ring is there, with there the ester part is attached, the further aromatic ring, with this aromatic ring NC double bond O C Cl Cl. So this is what basic structure, and the IUPAC name also given. So 4-chloro dichloroacetyl methyl aminophenyl furan 2 carboxylate. So this is what the IUPAC name of this dialoxanide. So which is what the directly acting amoebicidal drug. So mode of action of this is the dialoxanide furoate, which is what furamate, which split into the dialoxanide and furic acid. So this chemical structure is given over here. So of the dialoxanide furoate, so initially it split, means break down into the dialoxanide and furic acid. So furic acid death into the bacteria. So most of dialoxanide is absorbed, and the conjugates to be glucuronate, which is rapidly excreted into the urine. So most of these dialoxanide were absorbed, and their conjugates were excreted into the urine. And the unabsorbed dialoxanide, which shows the amoebicidal activity. So after the breaking of this dialoxanide furoate, which will convert it into dialoxanide and furic acid. Further, this dialoxanide into the absorbed form and unabsorbed form. So absorbed form were excreted as a metabolite into the urine, and unabsorbed form shows the amoebicidal activity. So this is what the basic mechanism of action of this dialoxanide. So the second drug, the further drugs from this category is Iodoquinol. So this Iodoquinol, which is what the quinoline ring system. So you can see the structure given over here. So basic structure is quinoline at second, third, fourth, fifth and seventh position there is iodine, and at eighth position there is hydroxyl group. So this iodoquinol is basically what the quinoline ring derivative, which is what amoebicidal drug. So effectiveness limited to the bowel intestinal organism, that is not effective against the intestinal wall, extra intestinal tissue, trophozoites. So this drug is only basically useful up to the luminal area only, or the luminal side only, uh, because it will effective against the intestinal wall and extra intestinal tissue trophozoites, which is not shows the activity against the trophozoites which were already penetrated into the intestinal wall. So the activity is only related to your bowel luminal organism. So the poor absorption of this drug is there, 90% unabsorbed drug. Renal excretion basically following glucose, whatever the glucuronic acid conjugation will be there. So in excretion we will get the glucuronic conjugate of this drug. So it may interfere with the sum of thyroid function test for up to the 6 month by increase proteins bound serum iodine which exceeded the iodine uptake, 131 iodine uptake. So this is what the function of this. So it interferes with the whatever the basic function of thyroid function. The side effect of this drug is on long term use it will causes the goiter, because we can see the into the structure there is iodine will be there. So goiter will causes, again the neurotoxicity will be there, potentially severe, and also the green stool will be there. So these are what the three basic side effects of this drug is. Note the green stool is indication of iodoquinol therapy. So whatever these kinds of appearance will seen into the uh, stool basically, so it means for the iodoquinol therapy is given. So further, so whatever chemical interactions, so optic neuropathy will causes, renal diseases, whatever the thyroid diseases, hepatic diseases, and other secondary amoebic size. So this is what about iodoquinolone. The next drugs from this category is the drugs which will effective only in the tissue or hepatic amoebiasis. So in that category the chloroquine and imetin is given. So these drugs we will discuss into the next session. uh, I hope all of you get these points. So till that, thank you and happy